Hello, my name is Bradley Alisea. I'm from the Open Worm Foundation, and I'm going to talk to you today about generative divergent integration as a theory of the emergent connectome. The first network I'm going to talk to you today about are embryo networks. Embryo networks are derived from cell tracking data in the embryo, and it gives you this five-dimensional space, 3 dimensions of space, 1 dimension of time, and 1 of context. That context can be put to use by analyzing this point cloud of cell tracking data and applied to a typical C. elegans lineage tree where you have time, it unfolds in time, and then this I, which is the context, is the AP axial order, or the anterior posterior order. It allows you to do very interesting things with the data. In general, cell division allows us to grow these embryo networks in terms of the graph diameter and the local connectivity, but also with cell division, you get global modularity. So as you get differentiation and you get cell division, you get this bipartite structure, but you also get this global connectivity, which is very interesting. You can see the spatial connectivity here, and you can think of this in terms of an interacting between cells, like in terms of cell signaling or other types of proximity-based questions that you might ask. So this actually allows us to explore connections between the connectome and the embryo network in various ways. And we can see that the connectome emerges within the embryo in very restricted spatial locations. Now we can also integrate those networks, and that's what the rest of the talk is going to be about. We can look at this in terms of the adult worm, so we can see that the connectome is spatially segregated within different parts of the worm, and it's segregated within the embryo. And the question is can we integrate these two types of networks, the connectome and the embryo network? This leads us to something called generative divergent integration. So we have 2 examples here. One is of an 8-cell embryo where the cells divide and you have this bipartite network based on the thresholding criterion that we use. But as the cell expands in terms of the numbers of cells and you start to get differentiation, you start to get 2 networks. You get an embryo network and then you get a connectome. Now, if you see from this example, the connectome is the green cells. It's disconnected just on the basis of the connectome cells. But if you integrate it with the embryo network cells, you have you have some sort of global connectivity. And this, it allows you for a much richer picture of what's going on in development. We can do very interesting things with this type of methodology. We can look at connectome formation, for example, and assuming certain rules about connectome formation, we can actually extract strategies and test these examples against data of all types, synaptic connectivity, and then put it back in the context of the embryo network. This leads us to something called the density bifurcation model, which is where the process of increasing connectivity allows us to build these sorts of rules for connectivity. So one of them is that when the cells migrate and divide, connectivity increases. The second is that cell migration enriches local communities and cliques, and those are network science terms, which basically means that you have groups, functional subgroups within the network. The function of cells diverge, which is differentiation, and 2 interconnected networks can emerge. So you can have 2 modules or 2 interconnected networks that are working together in the same embryo. And then finally, interconnected networks provide weak ties or functional interdependencies between 2 modules. And these could be emerging tissues or some other set of systems within the embryo. And so this leads us to something called a hyperlineage tree, which is a hypergraph based on a lineage tree topology. So you can see that as this hyperlineage tree reaches 4 cells, it's still a single network. But as you reach 8 cells, then you have 1 germline cell and 7 regular embryo network cells. And you get up to 28 network cells in the embryo network. The next stage of rounded division, this network bifurcates into 3 connectome cells and 48 embryo network cells. And so this embryo network continues to grow, the connectome continues to grow, and cells can always go from the embryo network to the connectome, but never the other way around. And this network actually captures a lot of what's going on in development. And so in future directions, we want to take these images of embryos where there's a lot of cell migration, a lot of tissue formation and differentiation and shape changes, and turn them into networks that we can understand better some of these systems that emerge. Thank you for your attention.